what I'm going to talk to you about today is this idea that I've been kind of working for the past couple of years of like how can we really extract information about biological systems using these machine learning models. And so I just want to tell you a bit about my background. And so I'm a graduate student at Caltech, and I'm in the biophysics program. But I'm actually originally from Mexico. And so I was born in Cancun, and then I did my undergrad over here in Merida. And I also worked at UNAM with a professor called Ernesto Perez Rueda. So we were really studying things about these gene networks. And also while I was in my undergrad, I had the chance to go to Madrid for 6 months to study also biochemistry and biophysics. And so around this time, there was a point where we started to learn about the kind of history of artificial neural networks, and we learned things about, for example, the Hopfield network and like how simple computations can actually generate the learning process. And so, just out of curiosity, does anyone here has a biology background at all? Yeah. Okay. Great. That's awesome. And what are the other backgrounds do you have? Like you can just shout it out, and I'll just physics, industrial engineering. Thanks. Sorry. Philosophy. Awesome. Well, that's great. Yeah. Like I like to have like kind of mixed. Yeah, kind of backgrounds, but just to kind of get a sense of where I can direct the talk. So, yeah, like out of curiosity, does any one of you know who this guy is? Well, this was given that we're in Pydata and we have learned a lot of neural networks today. So this guy is Donald Hebb, and he was one of the pioneers in thinking about these artificial neural networks. And kind of the ideas that Hebb had was that learning could be made by a very simple process in which certain groups of neurons can like associate themselves into like these clusters. And so like, like computation or like memory and stuff like that can be triggered by just having a activation of a subset of a given group of neurons, and that's kind of the theory of associative learning. And with this in mind, I started to think about that there were like a lot of analogies with how neural networks and how gene networks actually work. And so just also out of curiosity, do you guys know who this guy is? Well, yeah, I was just kidding. Yeah, so kind of the idea of this talk, the central point is kind of this approach that we're going to take today. That is just saying that how can we learn the wiring of these genetic networks that are actually controlling how cells behave, basically using machine learning. And so this idea is actually not that new in the sense that it has been widely known that genes that are wired together, they most likely fire together in the sense that, for example, if two genes are correlated in their levels of, in their amounts in a given cell, they're most likely part of the same process, right? And so this idea will be the kind of core of what we will do today during the tutorial. And so just to give you a sense of what we're going to do today is just to highlight that, again, we're going to use this knowledge that we already have from biology, that is gene regulatory networks, and specifically we will then train a machine learning model, and then we'll try to go back and to kind of create this feedback between these two kind of networks, really. And so I just want to make a disclaimer at this point that this kind of approach is not the end of the story. And so to me as a scientist, really what we're trying to do is trying to understand how living systems work. And in order to do this approach is just kind of just another tool that will finally, well, ultimately guide us to like make experiments and actually learn about how these complex systems that are really biological systems actually are, what are the causal relationships and the generative processes that actually trigger this stuff. And so biology really demands quantitative models, and this quantitative understanding can only really guide us through really grasping this great complexity that we're facing. And yeah, so just to give you an idea of this remarkable complexity, I want to use ourselves as an example. And so in us humans, there are about 6 billion letters of DNA in each and every one of our cells. And so if you actually think about it, like just like the sheer and utter complexity that makes us ourselves is really encoded in this idea of the chromosome. And if you think about the chromosome, it's like this big structure inside the cell that has like a three-dimensional shape to it and has a certain architecture. And so certain genes will be more in the outward layers of the chromosome, and some will be actually hidden and will be only active at certain parts of development and so forth. And so if you think about the chromosomes, it's really this idea of how complex actually genes, genetic information is encoded in the cells. And so also I want to ask you, yeah, like, do you guys think that that the cells in your brain and the cells in your heart, for example, are the same? Oh, well, better said, the genetic material that's in your brain cells and in your heart cells, is it the same? Yeah, right. And kind of this idea is very interesting, but actually what gives each cell its identity is how much of each gene is present at which cell. And this kind of cell fate is normally kind of chosen at development, but really is this kind of the underlying control that makes this really happen is these genetic networks. And so we can think of the cell identity as kind of the wiring in which these genes are connected to each other. And so before I get into more details of genetic networks for you to get kind of a background on it, I just want to talk to you about a bit of how genes are made in the cell. And so this is kind of like high school biology. So, and again, like if you don't come from a biology background, don't feel bad, because actually this diagram is wrong. Well, it's not wrong, but it's at least not complete. It's not the complete story, but this is just to give you a flavor of how the basic idea of genetic information kind of flows in the cell. And so we can think of this kind of software, if you want to think of it about it that way, as the genes, right? And so the DNA is kind of the software in the cell, and then this software has to get kind of transformed into a protein, which will be the actual hardware that will make up the cell. And so, interestingly, in the early 2000s, when scientists were trying to read the human genome for the first time, we thought that after reading all of the genetic code, like that is all of the letters that are written in our DNA, we will get kind of the whole understanding of the human genome, right? And so actually what ended up happening is that we found out that only 2% of the letters in your DNA actually ends up getting into a protein. And so this leaves us with like a huge amount, this 98% of stuff that is kind of these non-coding regions of the genome. And it's really interesting because we're just about getting to understand what is going on in this part of the genome, because sometimes people think or people thought that it was like this junk DNA, but it's really not the case. And so I won't get into the details of that, but just to give you a sense of like this sheer and utter complexity that we're facing as biologists, and that we really have to use like this kind of a motivation of why should we use these machine learning tools to really extract information as we can. And so this is not only the case of complex organisms such as ourselves, but this also happens in simple things like bacteria. And so besides being able to read DNA, we can also write it right now. And so this is just a diagram of the kind of process that was taking place when scientists made the first synthetic cell. And this was done just a couple of years ago at the John Craig Venter Institute. And this diagram is just saying that you start with a sequence of DNA. Sometimes it was a template. In this case, it was a bacteria. And so you then kind of make certain variations to it. And kind of the end goal was to make what was in concept a minimal cell. So what are the minimal genes that one needs to put into a cell that will actually live and reproduce, okay? And so what they did is they made a bunch of combinations, and then they ended up with a final minimal cell. And what was most interesting about it, and I just want to give you a flavor of this, what I like to call as biological mysteries, is that despite that they had a recipe to make a completely synthetic cell made of chemical constituents, almost 20% of those genes, like scientists had no idea. We had no clue of what these genes were coding for. We knew they were essential because if you took them out of the cell, of the recipe, you wouldn't end up with a fully baked cake. But in a sense, we didn't really know what the ingredients were in the first place. And so this, I think this is very, very interesting. And so what I want to claim now is that if we have kind of the wiring of of how these genes are connected to each other in this synthetic cell, we could have really had more of an idea of what is really going on with these so-called unknown or hypothetical genes. Okay, and so I just want to switch gears back to this idea of genetic regulation right now. And so kind of the main actors that are really controlling how the wiring takes place in the cell are these proteins that I really love, that are called transcription factors. And the story of these proteins is that they actually love DNA. They just are always trying to bind DNA and, like do their thing. And these proteins are composed of two modules. So one module, as I've said, is DNA binding domain. This basically, and sorry, there will be like a bit of technical jargon. So if you guys have any question at some point, like please just like stop me or raise your hand, whatever. Because yeah, like I'm kind of biased in the sense that I'm assuming that some of the words you will kind of understand, but I really want to get the message across. And so, as I was saying, these proteins are composed of two modules. One is a DNA binding module, and the other one is like so-called sensor domain. And this sensor domain is kind of receiving signals from the cell. Either they could be chemicals such as metals, or they can also sense pH, for example, or high temperatures or whatever. And this will dictate when they will bind to the DNA and control gene expression. And if we go a bit into the abstract for the kind of engineers, physicists, and computer scientists in the room, we can abstract these interactions into a directed network. And so here I'm just showing you this transcription factor that is controlling a so-called target gene. And so then these interactions can exert a certain function in the cell. And so, for example, what we like to call genetic circuits is these simple relations that are made by a few transcription factors and a few target genes, and they can, for example, elicit, for example, an oscillator in the cell. So the circadian rhythm in us humans that really drives when we are sleepy is basically controlled by a genetic circuit. And then if we could go to an even higher level of complexity, genetic circuits then form gene regulatory networks. And that's what we're going to talk about today. And we're going to use a simple case study that is kind of what I've worked on for these past couple of years, and is bacteria as a simple model system for studying gene regulation. And specifically, we're going to use this lab strain of bacteria that's E. coli K12, and we will use this because there's a lot of known information about it, and it's publicly available. And moreover, we're going to use as a case study, like even more specific, like this biological process of how purines, that are certain molecules that actually make up DNA, how purines are controlled in the cell. And so purines, interestingly, because they're essential to making DNA, they were really created in early stages of evolution. And so this is a very important protein that is called pure R. Very important protein throughout the bacterial kingdom, and we're going to use it as our case study for today. And so what we want to really do is to learn how the gene activation patterns of this protein is encoded into the cell. And what we're going to use as our data set is this so-called RNA-seq data that allows us to quantify how much of each gene is made in the cell. And so here this RNA-seq is just kind of a term for RNA sequencing. And so basically the idea is that we can read how much RNA is produced in a cell. And so here in this graph, I'm just showing you a plot of two genes in different expression conditions. And so in this case, it's just like an idealized example of two genes that are correlated. And what I want to also say is that this is not always the case, and there are very highly nonlinear interactions in the cell that are not really of this sort. But yeah, we will use this data set to, in the end, do this. We're going to take the gene regulatory network. We're going to build a binary classifier to predict if a given gene is inside this purine module or if it's not. Okay, so that's kind of the idea of this tutorial. And so yeah, so let's just go straight to it. So this is the link for the GitHub repo, and if you guys just click this or type bit.do/PydataLA, you will go directly into the repo, or you can just yeah, write it completely. So as I've said, if you go into the repo, you'll find this Colab and binder badges. If you click on it, I will use Colab just to be with because there are some changes that have to be done in the code. So yeah, if you're in Colab, you just have to uncomment these first couple of lines. So is everyone in the notebooks already? Everyone ready? Okay. So in this first, just to give you a sense of what we're going to do first, we're going to first extract the information from this purine metabolism simple gene network using publicly available data. Um, so first I'll just, it will tell you to, yeah, sorry. So it will do its thing, install some libraries and stuff. So the first thing we're going to do is that we're going to take data from this website. I will open just to show you while this is loading. This website called RegulonDB that is kind of a database of E. coli. And here in downloads, you can actually see this, if you go to experimental datasets. So if you're interested later on like continuing this sort of analysis, there is a bunch of data that you can actually download. So for example, the sequences, the genetic sequences, the genes, the gene regulatory network, and so on and so forth. So it's a very very cool so-called knowledge base of E. coli biology. And so we're going to use this data, and this website, RegulonDB, is maintained by the Collado lab at UNAM, and there are other kind of databases. So what I want to show you today is kind of this principle that we can take a data set and turn it into like a machine learning model using biological data, I mean. And so there are other so-called knowledge bases, for example, for Bacillus subtilis, that is another bacteria, for the worm C. elegans, and so on. You can also find data from humans if you want. There is a bunch of data out there. So I just will uncomment this, and this is only for the people that is running in Google Colab. It's almost there, but yeah. So what we're going to do first is we're going to load this genetic network of E. coli. And so if we look at just the first 5 rows of the data set, you can see that there's this column called TF, that is the transcription factor, the regulator, the target gene, this TG, the regulation type, if it's repression or if it's an activation mechanism, and other things like the evidence and so forth. And so the first thing we're going to do after is we're going to turn this pandas data frame into a network X graph. And so we will just use this command from pandas edge list, and it literally just takes a pandas data frame and kind of the columns that represent the wiring of the network. And so one of the ideas that one might ask is that, well, what is the kind of organization of this network? Like, we don't know anything about E. coli biology, and we want to know how is it organized. Well, one thing that you could ask is, who are the main actors in this network? Who are the main controllers, or so-called hubs? And so we can do this by just calculating a centrality measure that is basically getting the hubs. And so this is a method in network X called the eigenvector centrality. And so if we print the first 10 hubs, we will see that all of these genes pop out and their corresponding centrality measures. And so these genes actually are transcription factors, and they're also called the global transcription factors. And so these global transcription factors are basically master regulators of gene expression in the cell. And in this sense, with just a couple of commands, we can start getting important information about the architecture and also kind of the hierarchy of this network. And so yeah, if we actually sort this dictionary, we will actually get the highest scoring global regulators. Okay, and so yeah, now what we're going to do is we're going to take the initial data frame and just get which like from the transcription factor column which have the more counts in the data set. And what we'll see is that, for example, CRP is the one that has more interactions, and this is the one that we actually end up seeing as the top hub in the when we calculate the centrality measure. Now let's go ahead and start exploring this more in a visual way. And before we, what we want to do is kind of plot the network to see kind of its architecture. And so first thing we're going to do is we're going to extract the largest connected component, because you could imagine that in this kind of graph, there will be some kind of islands that will be just hanging around there. We just want to get the largest connected component that is the biggest part of the network that is completely connected, and that's what we're going to use as our visualization scheme. And so there is this simple command from NetworkX also that is based on matplotlib that we can just call directly on the graph object, and yeah, we will get a plot of the whole network. Okay, so this is kind of cool. And so what we can see is from this is that it basically is this exploding fuzzball. And one thing to notice, for example, is that there are these genes that are kind of making these ramifications. And so I assume that these are actually transcription factors that are controlling a subset of for example, a metabolic pathway. Okay, and so yeah, kind of the structure of biological networks in general is that they have a so-called scale-free structure, and that is that a lot of nodes have very few connections and few nodes have a lot of connections. And this small subset of nodes that have a lot of connections are the hubs. And moreover, these networks organizes into groups, and so we like to call them clusters in biology. And so is this kind of two properties, the scale-free property and also this clustering, natural clustering that happens in biological networks. Okay, and so the next thing we're going to do is that we're going to plot how many interactions each node have. And so this is kind of abstracted in the degree distribution. And so for each node, we will get how many interactions it has in the network. And we will use this library called bokeh-catplot, which was made by a professor at Caltech called Justin Boyce. And the nice thing about this library is that it allows you to visualize categorical distributions in a very nice and interactive way using bokeh. And so first we will make this helper data frame, because it uses a data frame as a source, tidy data frame. And so here we just have the name of the gene, which is the node, and the degree distribution, and if it's a transcription factor or a target gene. And so we can just go ahead and use this library, and we will make a strip plot, and we will separate the nodes by either transcription factor and target genes. And what you can see is that basically if you zoom in, you can see the annotation of each node. And so you can see the gene name and yeah, whether it's a target gene and so forth. And you can see that the target genes just make this cluster in like they have very few connections. And transcription factors are actually just having a lot of connections, which is kind of what we would expect. And if you kind of draw a hard boundary above 100, that's what I will call the global master regulators. And so all of these genes actually code for global transcription factors. And if you're interested, just as a side note, we will get into that, but you can use, for example, this website called EcoPsych. Because at this point you might think, okay, well, but what is this gene in the first place? Like, I don't even know. So let's just use CRP, for example. And with this, it's kind of like a Wikipedia of E. coli. And so you will see the gene, kind of what it does. And yeah, you can get a lot of information out of it. So yeah, if you're interested in just like finding out more about this data set, there's like a lot of information out there in EcoPsych. Okay, and so what we can see is that, okay, we're already kind of extracting the structure of this network. We've seen that there are very few genes that are actually controlling most part of the network, the so-called master regulators. And yeah, if we actually wanted to visualize it in a more interactive way, this graph, there is this library called HBplot that is kind of a higher level functionality than HoloViews. And there is a NetworkX module that we can use. And it's really, really high level, and it's really amazingly awesome. And yeah, we can visualize the network in interactive plot, and we can just hover around every node and see which gene is connected to and so forth. I don't know if this is taking a lot or... Yeah, it probably doesn't work in Google Colab. But yeah, if you run it locally, you will find out that it's basically the same visualization as before with Matplotlib, but it's interactive. All right. So at this point, another interesting thing is kind of exploring this clustering of this gene regulatory network. And so we can just use directly the graph that is the connections to kind of extract the clusters in it, the natural clusters that appear in the network. And so there is this algorithm for community detection in networks called the Louvain algorithm. And it's implemented in this Python library called community. And so we can just import it, and the method to call it is called best partition. And so in this object, it's already, you can already see the kind of the clusters that we have. Oh shit. I remember, I think this is a dictionary, sorry. Yeah. And so we can extract how many clusters we have, for example, in this network. And yeah, we have 16 clusters. So after extracting the clusters, I know this is very abstract, but we can map back the clusters to the network. And so we can also use this setNode attributes method from NetworkX, and we just give it the network. We give it the mapping, that is the communities, the clusters, and we will name this as modularity. And then we can iterate over the nodes in the network to get the cluster of each node. And we will then store this into a list. So this is kind of a convoluted list comprehension, but in the end, what we'll have is just for each for each node which cluster it belongs to. And so, for example, in this cluster, we have a bunch of genes, and we can then just go back to ecopsych and see which function they're encoding. And yeah, and so there's this little exercise that I leave for you later if you're interested. And so, so there's this other data set. So we took the whole gene regulatory network of E. coli, but we can also get only the interactions that are from the transcription factors. And so this you can download directly from RegulonDB in this called TF-TF interactions. And so kind of the question that I'm posing here is just if you want to cluster only the TF-TF network that is the kind of central control in this organism, you can actually do so pretty easily with the code that we already written. And so, okay, now is the main part. So what we actually wanted to do from this network is just extract the purine metabolism genetic network, right? And so we'll just simply filter over the data frame that we had in the beginning, just extracting the interactions that are encoded for this pure R protein. So we do that here, and then we're just going to check how many genes are controlled here, and we can see that there are 31. For example, you can see here. And so all of these target genes are being controlled by the pure R transcription factor. And then we can save this as a CSV, but it's already in the GitHub repo, so you don't actually have to do it. And then you might think, well, then how are we going to even make our training and test set in the first place? Or how can we really know if we are actually predicting something that is biologically relevant? Well, the nice thing about this is that there's this other study by the Paulson lab at UCSD that has already done this thing experimentally. So we will use that as our kind of gold standard and see if we can actually get the same results that they got experimentally using this machine learning model. And so if you're in Google Colab, you also have to uncomment these lines. Okay, and so it's basically the same structure of the data set. You have the transcription factor and the gene that is being regulated by it, and if it's an activation or a repression. And so we're going to do the same. We're going to filter out only the purine gene network that is also called Regulon. So if you hear me saying Regulon, it means gene network. And yeah, we're also going to save this as a data frame, and we'll be good to go. And that is going to be our training and test set. Okay? Cool. So now, does anyone have a question up to this point? Yeah. Yes. So we will get into that in a moment. So one idea that we will use is that we will use oversampling to enlarge the data set. So kind of learn the distribution of this small subset and then use that as our training. But it's a good question. So yeah, that's kind of the idea. Like, we'll have a lot of negative examples that are genes that are not inside this subnetwork of purine metabolism. Like, you can have genes that are coding for the cell membrane that is making fatty acids, or genes that are making amino acids, or genes that are making, I don't know, DNA repair or whatever. And yeah, that's why we're going to have to use oversampling. So let's go to the second notebook. And so this is where the core of this tutorial is. We will take the expression data set and we will train our model using the wiring that we just extracted. So if you're in Google Colab, also you have to uncomment these lines. And so while this is loading, I can tell you a bit about this data set. And so this data set was also published by this Paulson lab at UCSD. And basically, the data set contains, in each row, it's a matrix. And so in each row, it's a gene. So in the E. coli genome, there are about 4,000 genes. So there are like 4,000 rows. And then the columns in the data set represent different experiments. So they grabbed E. coli, they made a culture of it in a big flask and so forth. And then they started making perturbations. So for example, they grew it in different sugars. So they feed E. coli with different sugars, or they deleted a gene and see what happened to it. And yeah, there are like about 50 expression conditions in duplicates. And so this is kind of the structure of the data set. Is basically each column represents an experiment of the expression of all the genes in different conditions. And then the basic units of like the entries is in this measure called transcript per million. And it's basically a proxy of how much of a given gene was made into RNA. Yeah, and so this was a very cool paper. So here I'm leaving you the link. So while this loads, I'll just talk about a bit about the paper. So they were also interested in this idea of like unknown genes. And so remember I told you that that in this synthetic cell, about 20% of the genes we really had no clue about what they were doing. Well, even in simple E. coli that we have been studying it for more than, I don't know, 50 years, a lot more than that. There are about 30% of the genes that we don't have a clue of what they're doing in the cell. And so a lot of this is just a lot of problems in like database information and stuff. And so what they did in this paper was just trying to extract information from different databases and kind of making everything into like this comprehensive thing. And one of the things that they found is that basically these genes that we haven't really discovered is because they're present in very little amounts in the cell. So they're really not causing a phenotype that we can actually measure. And so that's one of the ideas that they have into why is this thing so difficult in the first place. Okay, so let's just go ahead and load the data. And just look at the shape. So as I've told you, there are like a little more about 4,000 genes. And if we look at the first five rows, we'll see that there's a column of this annotation called locus tag that is basically just an ID for each gene. Then we have the gene name. As I've said, each row represents a gene. And each column represents, for example, here they grew the wild type, that is the kind of the normal E. coli in glucose. then they did, for example, a mutation and so forth. And so these are just a bunch of expression conditions. For example, different, like they put some acid and see what happened to the cell and so forth. And so this is kind of the structure of the data set. Oh shit, what did I do? Okay. And so here I'm just going to make a copy of the data set, and then I'm going to take the first two columns, that is the locus tag and the gene name, and then we're going just to take the actual numerical values as this data, sub-data frame. And so first things first, we're going to normalize our data set using a simple standard scalar. And then we're going to see if there are any null entries or null values in our data set. And so here we can see that it's just like a mean of 0 and a standard deviation of 1. This is just a z-score. And yeah, we see that there is no missing entries, so we're good to go. So the next thing is just loading the data sets that we generated in the last notebook to start making our annotations for the output of our classifier. Right. Sorry, you just have to add quotes on this URL. And same thing for here. Sorry about that. For the Google Collabers. All right, so we have our data sets loaded. And yeah, we have in our genetic network, there are 4,000 nodes. Yeah. Yeah, so the samples are actually the genes, and the features will be the expression conditions. So in the last visualization was just like kind of transposing the, so in the presentation that I show you like the scatter plot of the genes, I just made a kind of a transpose of that. Not in this case, but just to make the visualization. We'll get into that, but yeah, so it's a good question, but yeah, basically the samples will be our genes, and the features will be different expression conditions. Okay. Yeah? All right, so just remember we have 31 nodes in this pure R sub-network. And so what we want to do is we want to get the target genes, that is the genes that are controlled by pure R. And we'll just extract that as a NumPy array. So from the data frame that is of the pure R subnetwork of RegulonDB, we'll take the target gene column, and then we're just going to take its values. So you just have to, for the guys at Google, who's using Google Colab? Okay, so most of them. Okay, so you just have to go to the repo. Okay, and then under the data folder, you'll find this high TRN CSV. So I think that's the one that we're missing. Yeah, exactly right. Thanks. So you just have to change this URL and basically put the high TRN one. Thank you for the one who noticed. All right, so we should be good to go. So you have to put this, URL pure RDB variable under this read CSV command. Do you guys see it? And so now we should, okay. And yeah, so as we knew, there are 31 target genes. And we can also see this just like to see like the actual gene names. So all of these, and if you look, for example, these other proteins called pure A, pure B, pure C, this is like the set of genes that are actually making the synthesis of purines. And these other pure are the ones that are making pyrimidines, that are other molecules that make DNA. And so in this sense, this is just to give you a flavor of the actual genes that we are handling here. Okay, so now we should be good to then take from the Paulson data set that will be our test set. And we're just going to basically get the set complement of the intersection of these two data sets, just to get the genes that were discovered by the Paulson lab. So this is the genes that we're going to try to predict effectively using our machine learning model. So is everyone following me at this point? Is there any question? I see kind of a... Everyone good? All right. And so now I just want to give you more of a sense of how is this data set actually composed. And so what we're going to see is this just very simple scatter plots between the genes. And so this is kind of what I showed you in the slides before. And yeah, we're going to just take all of the genes that are inside the our test set, these 5 genes, and we're going to plot them against pure R, with the idea that if they're kind of correlated, then we might expect some biological relationship. And so here I just made a simple wrapper function that basically extracts the data for these given genes, and then makes a Seaborn scatter plot with the Pearson correlation coefficient embedded. And so we're going to loop over the genes, and then we're going to make these plots. Okay, so as you can see, some of them are, like for example, this one called peer I is very, it doesn't look pretty much correlated, right? But there are others, like for example, SHAM-P. This is for another, this is a gene that codes for the synthesis, I think, of another molecule called xanthosine, and this other important molecule in the purine pathway. And so we can see that this is more correlated and stuff like that. So now what I want to do is just use a simple principal component analysis to kind of use this as a noise reduction technique. And so the basic idea is that we're going to grab our data set, then we're going to project it into a latent space that has 80% of the variance of the whole data set. And then in this subspace, then we're going to convert back to our original dimensionality. And you will see that actually this forces the data set to become more correlated. And so, yeah, if you guys are interested in seeing how this kind of works, I leave this link to a very nice blog post. So yeah, let's just go ahead and do it. So we're just going to call this PCA object. And so just to get a sense of kind of the dimensionality of our data set. here we can see that we're getting, like, in just a few dimensions, we're getting most of the variance, 90% of the variance. And so we can see that this data is actually very low dimensional. So just by taking the first 5 components, we get actually 94% of the variance. So we're going to use that. We're going to project into this just 5-dimensional subspace. And then we're going to convert back to do this noise filtering procedure. So in this sense, we just transform into the subspace, and then we convert back using the inverse transform method. And we can see that, yeah, we go back to the same dimensionality. And then I'm just going to add the annotation to this reconstructed data set, add the annotation of the gene names to this reconstructed data set, and that's what we're going to work on. And so now if we do the correlation plots again, and if you go back and compare, we will see that actually we're forcing the subspace, because we're actually projecting to this subspace that has just like the most of the variance in the data set. We're forcing the kind of reconstruction to have more covariance between the features. And so this is just to show you that this is just going to be a kind of a noise filtering technique that we're going to use. All right, so to continue, let's just visualize how our dataset looks in the PCA space. Oh no, Holobius is not working in. Damn, this is not working in Colab. I haven't noticed, but damn, this is so bad. Yeah, it won't work. But yeah, kind of the idea is that when you run it locally, if you want, you'll see that there is basically this blob of genes. And so what I want to claim is that basically a nonlinear dimensionality reduction might work better for purposes of this highly nonlinear data set. Okay, so how are we doing? Is there any question up to this point? We're almost there, guys, almost there. All right, let's just continue. I'll just take a deep breath. All right, let's continue. So, yeah, so now we just have to annotate our training and test data set to have like the output of our classifier, as I've said. So this is just a wrapper function that will actually extract data from a data frame given a gene list. So we will use the list that we made before from the pure R regulans from RegulanDB and the Paulson dataset. And yeah, so before we do that, let's just add this as a one-hot vector. And so what we'll do is to say that if the gene is inside this in this set of genes of the pure R gene network, we're going to put a 1, and if not, we're going to put a 0. So this is kind of the one-hot vector. And then we will add this one-hot vector into our data set, this noise-reduced data set. So what we'll end up with is this data set as before. Just in the end, we have this. This will take a long time. But yeah, in the last column, we will have the annotations of if it's inside or outside our gene network. And so, yeah, we'll use this wrapper function, as I've said, to just extract the test set. So if you can see, it's just these genes that we've already visualized, SHAM-P, peer I, peer B, and so forth. So we're going to eliminate them from the reconstructed data set. And then, because we want to see also if the network really finds out about the false positives, let's just sample from this, from the genes that are not inside the network. We'll sample without replacement just to make this kind of a noise and add this to our test data set. So in this sense, we'll make this kind of unbiased test set. And for the last notebook, remind me to use binder because I really want to for you guys to see the interactive plots. All right, so now I'll just make a copy of this unbiased set. And yeah, we'll see that it has a bunch of other genes that are not inside the pure R regulon. And if you think about it, these genes might be inside the regulon because that's actually what we're going to want to predict. You see? It's like we're trying to match the experimental predictions of this Paulson lab. But if we wanted to actually discover new genes, we could use this model. So we will just use as kind of a validation that the method works, but these genes actually might be in the regulon, who knows? All right, and then just make this training subset, just making a copy of the other. All right, so now we just have to split our data set and yeah, just checking the dimensions that we have. We have 35 genes in our test set and 4,252 as our training. And so, yeah, just the output will be the last column, as I've said. So here I'm just selecting the data and the output. And it's here just checking that is this for the training is just numerical values, and for the output is for the X data set is just numericals, and Y is just a one-hot vector. And so same for the test. All right, and so last thing we have to do before proceeding is, for example, if we look at the training data as he already mentioned, is like we have very few data points of positive examples. So we have to do oversampling. And so there's this very nice library called Imbalance Learn that has a bunch of implementations to do over and under sampling. So because we have so few, we will actually use oversampling. And there's this technique called SMOTE that is based kind of on this k nearest neighbors. It's kind of making this data set using nearest neighbors as a distribution. Sorry. So we will use this method to make the oversampling. And so with this simple command, we now have kind of a balanced data set in order to make the training and test. All right. Yeah, so we can just try a couple of models to see which one works best. And so just for now, I want to say that we will start with very simple models and then kind of build up on complexity and see which one works and which one doesn't. And so first we will use a a linear support vector machine. And let's see how well it predicts. Okay, guys, are you ready? Does this thing work? All right. Well, we can see that it's not bad. Let's see at all of the metrics. I would say it's pretty decent. So, in this sense, what we can see is that, yeah, and if we just look at which are actually, yeah, we basically get a pretty good prediction. I don't know if this is a gene that was actually in the Paulson dataset, but we did pretty good. And so what I want to say now is that at this point, because this is a perhaps very small subset of genes, a very simple genetic network, I will be happy to just continue just doing hyperparameter tuning and just keep this linear support vector machine. But let's just try other models to see how we roll. So for example, if you use random forest classifier, well, Ediboost is kind of like a ensemble model also. So let's see. Also pretty good. We were kind of better with the support vector machine. So this might be what I think is that this might be perhaps too complex of a model for our data. So it might be actually overfitting our data. So in this sense, I will trust the linear SVM. But just to kind of do what I promised and use some neural networks, let's just use a simple Keras model with one hidden layer to see how well we do. Yeah, and there's this weird. I think I have an older version of Keras, but sorry. Yeah, so just in the last step, it's kind of 94% accuracy. So yeah, I noticed this error in binder, but yeah, I won't worry about that too much. So yeah, I'll just do some simple cross validation to make sure that the support vector machine is actually doing decent. And so yeah, it's pretty good model. So let's just wrap it into a pipeline that basically takes our training and test data set and just scales the data and performs the classification. Yeah, and so at this point, if you want to, how are we on time? If you want to actually use this model, I think you can export it now and just start making predictions about this simple genetic network. Okay, and so, all right. I think we have just a couple of minutes, right? 15 minutes, okay. What do you guys think? Like, we can just take some questions and talk for 15 minutes, or if you guys, yeah. Yeah, so, or the other alternative is just, we can do a simpler exploratory data analysis in a kind of unsupervised way, just to kind of get a flavor of the data set using nonlinear dimensionality reduction. Let's just take your question and see.
So for a completely unseen transcription factor,
yeah,
you would have to run those 124. I'm sorry. So for a completely unknown transcription factor,
yes,
you would have to rerun those 124 experiments.
That is a very, very good question, actually. And that is actually the kind of basis of my thesis, my undergrad thesis. And so, yeah, I think at this point is, there are like 1/3 of the transcription factors that we really don't know what they're doing. but like we know that it binds DNA and they have a sensor domain and so forth. And so what we did is just kind of an unsupervised learning, the thing that we're going to see next. But yeah, it's a very good question. And what I would say is that if I understood your question, yeah, what I would do is just stick to the experiments to see, kind of get a better flavor of what these genes are doing.
Well, if you are going into the machine learning world, right?
Yes.
You can also find feature importances. So perhaps you could, find the most important experiments to conduct, right? And get your decision. That's also another, something you could maybe look into.
Wow, that's a great idea. Yeah. No, thank you. It's really great. Yeah, never thought about it. Yes, it's this thing of like, what are the, yeah, exactly what you said. Like, what are the experiments that will give us the most information out of these biological systems, right? I think that's a very good question. More questions or ideas? I don't know. Okay, so yeah, let's just take this 15 minutes, if that's okay. And if other questions, you can just stop me. So I'll just switch to Binder because it's heavily visual. I don't know how much this will going to take. Oh yeah, this one is broken already. All right. Ah, shit. Yeah. I think I tried it in Binder, so it should work. The weird thing is that bokeh catplot did work, so I don't know, maybe I messed up something with whole views. All right, so basically the idea here is that biological systems are highly nonlinear. So, for example, even a simple chemical reaction, an enzymatic reaction, involves a nonlinear parameter. And so, in this sense, I think that nonlinear methods will really help us in uncovering the structure of our data. And so here I just make a rant of what I think about this in the biological realm. But basically, the idea is that there's this nice method called UMAP, and it's implemented super nicely in a library called UMAP-Learn. And if you want to get into the details, I also leave a blog post here. But yeah, let's just go back and load the data. And I'll leave this thing running and keep on talking. And so basically the idea is that UMAP will also find kind of like this manifold of that captures like the global structure of our data set. And so in this sense, we'll just use manifold in 2D. That makes any sense. And yeah, we're going to then just try to visualize it if we get some structure out of it. So here I just add just these 2 dimensions to our data set. And yeah, let's see what we get. Oh shit. It's this weird kind of croissant. And so what is this actually telling us about biology? Well, I guess that we have to find out. Like, there has to be this hidden variable that's making this variation, right? So yeah, like one idea is just like do some clustering on the data. And yeah, you might say that, well, there's not any clusters in that weird blob, but we'll just use clustering to like actually extract the things that are in the in the edges of this croissant. And we'll just use a Gaussian mixture model to do it. So this should work in no time. And the nice thing is that it's a probabilistic model, so we actually get probabilities for each gene. But we can also get the most likely cluster that each gene will fall into. And so let's just quickly visualize it. Yeah, and so this is kind of neat. So we see that cluster 4 is over here. And I thought this was going to have a reproducibility to it, but it seems that it changes from batch to batch. But yeah, basically, the idea from here is to extract the clusters in the edges and see if there is like an enriched biological function. So kind of the idea, if we had more time, would be to like go cluster from cluster and see which function is actually enriched in each kind of subsection of the latent space. And so these are not the edges, but let's just go with it. So we're just extracting, in this example, cluster 6 and cluster 11. And to get the enriched biological functions, you can just use this annotation-based thing that it's kind of the golden standard. It's called gene ontology. I don't know why these links. So one database that you can use is actually called Panther. And what you input is just a list of genes, basically. And it will, sorry, this is, you're not seeing this, right? Yeah, you basically give a set of gene names, and it will output what functions are overrepresented in the dataset. So what I did is I downloaded the annotations, and I made like some simple wrapper functions. And so, for example, in cluster 6, there is nothing enriched. And then in cluster 11, no luck either. Oh God, it's awful that changes from batch to batch. Well, I had no luck in this time, but basically, you can go cluster from cluster to cluster, and I made this like simple hypergeometric test in this Py file that I'm putting in the repo, and you can see then in EcoPsych just which genes are like getting more of a story of what you're getting. So yeah, this is it. I just want to thank you for paying attention, and just tell you again that this was based on work that I did in the Perez Reda lab at Mérida. And yeah, if you want to collaborate or just talk more about these ideas, feel free to reach out. And yeah, GRN stands for Gene Regulatory Network, if you have noticed. Okay, so thank you guys.